Welcome to chapter 3.4's video. 3.4 is all about stains and how we make microscope slides. Chapter 3.4 is probably one of the most important sections we talk about in microbiology because this is going to apply to the class and to the lab equally. So a lot of stuff that we're going to see here, we're going to actually be doing hands-on in the lab. But before we start into the material for 3.4, let's do some review questions. Okay, review question number 1. Which term refers to a lens's ability to produce clear, non-blurry images of a microscopic object? A. Magnification. B. Amplification. C. Resolution. Or D. Scattering. Okay, take a second, pause the video. Let's see if you can figure out this answer. Okay, the answer for this question is C. Now, we haven't talked about amplification. This is a characteristic of waves, but that we talked about back in physical science, but we haven't talked about it now. So the only three that we've talked about in class are magnification, resolution, and scattering. Now, magnification is making an image larger, so that's obviously not the answer. So it's going to be either C or D. Now, scattering means that everything goes in all directions. You actually don't produce images if light is going in all directions. So the answer is C, resolution. Okay, review question number 2 time. So the ocular lens of a compound light microscope is the lens that A, is closest to the observer's eyes, B, is closest to the object being observed, C, focuses the light from the illuminator on the specimen, or D, the light from the illuminator passes through first before reaching the object. Now, just to be aware, illuminator is another word that we use for light source. Okay, so this could also refer to the lamp of the microscope. So take a second, pause the video, and try to find the answer to this question. Okay, the answer for this question is A, is the closest to the observer's eye. So ocular means eye, so it's the eye lens or the eyepiece lens. Now this is actually a really good question to know because this is an example of a question that can be rearranged on an exam to ask for different parts of the microscope. So if it was asking for the objective lens, then the answer would be B. If it was asking for the condensing lens, that would be C. And if it was asking for the actual condenser, the answer would be D. So all of these refer to parts of the microscope, but A is just the one that is correct in this case. Okay, chapter 3.4 is all about stain samples. We're going to talk about how we increase the contrast of our microscope slides. Okay, so our objectives are explain how a stain reveals additional information about a microscopic object and be able to describe how the gram stain distinguishes two classes of bacteria. Now, before we start in on actual staining, let's do a case history. And this is blood slide reveals a parasite. So this case history is all about Purna. Now, Purna is a 6-year-old boy that lives in India. So the part of India where Purna lives is experiencing monsoon season. So it's really rainy all the time. Well, this monsoon season is even rainier than normal. Okay, so much so that water is starting to pool in areas all over the ground. Now, one day Purna wakes up and he's not feeling too good, and he slowly begins to experience symptoms that include a high fever, intense chills, like he can't get warm. His chills are so bad that he's shivering and shuddering all the time. He has intense headaches, like migraine level headaches, and he's also nauseous and vomiting. Now, because it's an extra rainy monsoon season and there's pools of water all around, this is not good for India. India is a very warm climate. So anywhere that they have standing water, they're going to start to have mosquitoes using that standing water to breed and make more mosquitoes. The same thing happens here in Idaho. Well, Purna's symptoms are going to subside, so his fever is going to break, and then it's going to return 3 days later. Okay, and then this cycle is going to repeat. It's going to go away, it's going to come back. It's going to go away, it's going to come back. Okay, so after several weeks of these cycling fevers, Purna's family are really worried. So they brought him to the clinic to be examined. Okay, so when they were going through the different tests, Purna showed that he had an enlarged spleen and liver, and it was extremely anemic, so he doesn't have a lot of red blood cells. So one of the tests that the doctors do is they take a sample of Perna's blood and they smear it on a slide and use what's called a Gemsa stain to image the red blood cells. Okay, so Gemsa contains eosin, which is a dark negatively charged dye, and this dye is going to bind to the outside of the red blood cells so we can see them more clearly. Now, if you look at red blood cells underneath the microscope with no dyes, they're clear and they're very hard to see. Okay, and then the second part of the Gemsa stain is methylene blue. So methylene blue literally dyes things blue, but it's a positively charged dye that's going to bind to negative charged particles such as the phosphates in DNA. Okay, so we're going to do that, and what we're going to see with Perna's slide is we see the red blood cells. They're going to look like this bluish purple, but some of them have these really dark purple rings in it. Now, this is very important. This is going to help us figure out what Perna has. Okay, these rings are very indicative of a very specific disease. Okay, so these rings show that Perna has the parasite Plasmodium falciparum. Okay, this is the parasite that causes malaria. Okay, so these dark rings that we are seeing in the red blood cells, this is the parasite. This is its nuclei. Okay, so what happens here, this is why Perna was anemic. So the parasite is going to enter your red blood cells. Now, when the parasite's in your red blood cells, your immune system can't detect it. Okay, this is when Perna's fever would break. Okay, so it would be in the red blood cell, it would be eating, getting all its nutrients, start to replicate, and so it's going to burst or break open this red blood cell. Okay, which pretty much destroys the red blood cell, and it's going to allow the parasite to go and infect a new red blood cell. Okay, this is why he's anemic, is because of this process of the parasite breaking open the red blood cells. He didn't have it enough to not feel tired all the time. Okay, so in Perna's region of India during this monsoon season, over 400,000 of individuals were also infected with malaria and became ill. Okay, this is very high numbers. In fact, malaria is actually one of the most deadly diseases worldwide, even still. And because we don't really have any treatments for it. We can give some medications that will help control the symptoms a little bit, but we don't have anything that will really allow us to kill the parasite, because the Plasmodium falciparum is a eukaryotic parasite. What else is also a eukaryote? Us. Okay, so we haven't really been able to find treatments that will treat it without hurting our cells as well. Now, a few years ago, they started to develop a vaccine for malaria, and I think it's in clinical trials. I'm not sure if it's ever become commercially available yet, but they are working on it. Okay, so Perna's case study shows us the usefulness of using stain as a microscopic method to diagnose disease. Okay, so what we're going to do as we go through this lecture towards the end, we're going to talk about how we make slides in the lab. So we're going to talk about things like fixation and what actually happens when we stain. And then we're going to talk about some specific stains that you will need to know for the exam. Okay, so let's start with the first part. So we have our sample that we want to look at, and we have our microscope slide. Okay, so what we're going to do is the first step. Okay, so we're going to get a loop. Okay, so we have these inoculating loops in the lab. They are literally a wire stick with a round loop on the end. Okay, inoculating loops are a way that we can move small amounts of a liquid from place to place. Okay, so we're going to take our inoculating loop and we're going to dip it into our broth culture, and we're going to get our sample out. And we're going to thinly smear this sample over the slide. Okay, let me get my pointer here. Okay, so we're going to spread it out. So we get it as thin as possible, and we're going to let it air dry. Okay, so once our thin film has dried in the air, what we're going to do is we're going to do what is called fixation. Okay, so this is going to be a process. It can be chemical or physical process that will kill the cells, and will cause them to attach or adhere to the sample slide. Now this is important because we want our samples to be stuck to the slide so they don't wash off in the staining process. Okay, so there's two main methods that we can use for fixation in the lab. The first is what we call chemical fixation. So we're going to use a chemical to kill the cells and attach them to it. The most used chemical is methanol. Okay, so we'd put a drop of methanol on there, spread it thin, and we'd let it air dry, and it's going to help the cells adhere. Now, we don't use chemical fixation in the lab when we do this class. We're actually going to use a different method, and it's called the heat fix method. Okay, so what we're going to do is we're going to get our slides, and once they're air dried, so once we're at this step here, we're going to take our air dried sample, and we're going to take it and we're going to kind of swish it over a flame from the Bunsen burner. Okay, so the flame is going to be used to kill the cells and cause them to stick to the slide. So this is what we call heat fixing. Now, we got to be really careful, okay, when we're heat fixing a sample, because if we get the slide too hot, we're actually going to destroy the cell shapes and not just kill the cells. So we could go and look and not get the right image. So we might not see what we're really looking for because we destroyed the cells in the fixation step. So this is a very important step, but also a step where we could have some misfortune, unfortunately. Okay, so we have heat fixed or fixed our sample to our slide. Now it's time for the contrast step. Okay, so if we just use a single stain, it's called a simple stain or simple staining. Okay, so staining is the step after fixation where we're going to add a chemical stain or dye to the slide to increase the contrast so we can see more underneath the microscope. Okay, so this is our procedure for a simple stain. Okay, so the first three are our fixation step. Okay, so we're going to put the sample on, we're going to make it a thin film, we're going to let it air dry. So in this procedure, they're going to add methanol and let it air dry to fix the cells to the slide. But we're going to use heat fixing here. Okay, so it doesn't matter what, these first few are always going to be the same step. Put the sample on the slide and fix it to the slide. Okay, now comes the staining part. So with a simple stain, we're only going to use one chemical or one dye to increase contrast. So we can use things like methylene blue, like in this example. We could use iodine. We could use a dye named saffron or a dye named crystal violet. And there's quite a few different stains, but those are our main ones that we use in microbiology lab. So in this one, they're going to put a few drops of the methylene blue stain on the slide. Okay, and we're going to let it sit there on the slide for 1 minute. We're going to give it some time to soak into the cell so we can actually have something to image. Okay, so when our timer goes off, our minute is done, we're going to rinse off our stain with water. Now, if we don't do the heat fix step at the first and fix our sample to the slide, in this step, when we're rinsing the excess stain off, we'd also wash our cells off the slide as well. Okay, but we're going to rinse the excess stain off. Okay, we're going to blot the excess water off. And then once it's air dried, we can use the slide underneath the microscope. So that is the simple staining simple staining step. We are going to be actually doing this in the lab. I think this is lab 2 or maybe lab 3. One of the first 2 labs after the measurement lab. Okay, so we will be doing this pretty quickly in the lab. Okay, so a simple stain is when we use one dye. Now, frequently we have samples that have a mixture of microbes in them or a mixture of bacteria. So we need to be able to stain them in such a way that we can tell the difference between the different types. Now, this is where a differential stain comes in. Okay, so a differential stain is a method of staining that's going to allow us to tell the difference between 2 kinds of cells. And it's going to allow us to see them and be able to separate them in our minds. Now, we have a lot of different types of differential stains. We're going to talk about a few in this towards the end of this lecture, but right now we're going to talk about the most famous differential slide. Okay, or differential stain on this slide. Okay, and this is important. We use this stain all the time. In fact, you guys are going to be masters of using this stain in the lab by the time we're done with the semester. Okay, and the most famous differential stain is the Gram stain. Okay, this is actually named after a guy named Gram. So the Gram stain is important because it allows us to distinguish between what we call gram-positive cells and gram-negative cells. These are both bacteria cells, by the way. Okay, so with this differential stain, it's going to allow us to see each type of cell, each type of bacteria, as a different color. Okay, so in the Gram stain, gram-positives are purple, while gram-negatives are pink. Okay, you need to know these colors. You need to be able to tell the difference between these. This will be something you're tested on in class and in the lab. This comes up all the time. Okay, gram-positive bacteria are purple, gram-negative bacteria stain pink. Okay, before we start talking about the actual procedure for gram stain, let's talk about the two types of bacteria it tells the difference between. Okay, so the gram stain is differential for gram-positive bacterial cells and gram-negative bacterial cells. Okay, so in the Gram stain, gram-positives are going to appear dark purple, and gram-negatives are going to stain pink. Now, both of these types of bacteria have cell walls. Now, when we talk about cell walls, we think about plant cells first, and the plant cells are going to be made up of the polysaccharide cellulose. Now, bacterial cell walls are made up of a polysaccharide, but it's not cellulose. It's actually called peptidoglycan. Okay, so the difference between gram-positives and gram-negatives is how much peptidoglycan each cell has. Now, gram-positives are going to have a thick layer of peptidoglycan, while gram-negatives are only going to have a very tiny, skinny little layer of peptidoglycan. Okay, this is what's going to allow the Gram stain to work. This is why it's going to be a differential stain, because it can help us tell between the two types. Now, let's talk about the Gram stain. So we have 5 steps in the Gram stain. It's actually more like 9, but we'll talk about, or 8, but we'll talk about that here in a second. Okay, so the first step, once we have the sample on the slide and air dried, we're going to do our fixation step. We're going to heat fix it. We're going to use methanol. We're going to let it air dry. Okay, once it's air dried and fixed, we can then start the Gram stain procedure. Okay, so here in step 1, after the fixation step, both gram-positive and gram-negative cells are going to be colorless. Okay, this is important. It's not going to have color until we add the stains. Now, the first step in the Gram stain is to add what we call the primary stain, or crystal violet. Okay, so we're going to put a few drops of crystal violet on the slide, and we're going to let it sit there for 1 minute like we did the methylene blue in the simple stain. Okay, once our minute is over, we're going to rinse the excess stain off, and we're going to blot it, even though the rinse step is not mentioned here in this flow chart. Okay, so after the crystal violet step, both gram-positives and gram-negatives are going to be stained purple. Okay, the second step in the Gram stain procedure is we're going to add iodine. Iodine is what we call a mordant. Okay, so it's going to help the stain stick to a specific type of cell better than another. So when we add the iodine, it's actually going to combine with the crystal violet, okay, in the gram-positive cells, and so the stain gets stuck in that thick peptidoglycan layer. Okay, whereas the gram-negatives, they have a thin layer. This complexation step, so where the iodine and crystal violet combine, doesn't happen. Okay, so what's going to happen is this is going to allow the crystal violet to stick in the gram-positives, but not the gram-negatives. Okay, so we let the iodine sit for a minute, have it complex with the crystal violet in the peptidoglycan layer, and then we're going to rinse off all the excess stain. Okay, our next step is what we call the decolorization step. So we're going to use a decolorizer. in this case, it's going to be the alcohol ethanol. And we're going to put that on our sample, and we're going to let it sit there for 20 seconds. Now, in the lab, we actually only use about 15 seconds because I find that when we set the timer for 15 seconds, and by the time you guys get things rinsed off the slide, it's been 20 to 25 seconds. Okay, so the decolorization step, the ethanol step, its whole job is to remove stain that's not fixed into the peptidoglycan layer. Okay, so with the gram positives, we're still going to be purple, right? We had the crystal violet complex with the iodine in this third step, and it's stuck in that peptidoglycan layer, so we can't wash it away with the ethanol. But with the gram negatives, we don't have a thick layer of peptidoglycan, so the crystal violet can't stick. Okay, so what's going to happen is the ethanol is going to wash away the crystal violet. So at the end of the decolorization step, we're going to have purple gram positives, and we're going to have colorless gram negatives. Okay, the last step is what we call the counterstain step. Okay, so we need to color these now colorless gram negatives so we can actually see them. So the counter stain that we're going to use is the stain called safranin. So this is a pink dye. Okay, so we're going to add our safranin to our sample, and we're going to let it sit there for a minute. When our minute's over, we're going to rinse it off and blot, and then our sample is going to be ready to view underneath the microscope. Okay, so our crystal violet our gram positives are going to still be purple. The crystal violet's still there. The safranin's there too, but the crystal violet's so much darker that we can't see the peak. But the important part, and this is the differential part, is this counter stain now has our gram negatives appearing pink underneath the microscope. So if you're ever asked what the most important step in the gram stains procedure is, it would be this decolorization step, because this is the step that makes this a differential stain and makes it so we're able to tell the difference between gram positives and gram negatives. Okay, you need to know this procedure front and back. Okay, you need to know what you should see at each step. You should know what dye or decolorizer is being added. You need to know the vocabulary. Okay, so that's where this slide comes in. Okay, this goes through everything I just explained on the previous slide. The primary stain is the crystal violet. So we have our fixation step. We have the crystal violet. Okay, we put it on for a minute, rinse it off, the excess off. We get both gram positives and gram negatives are dark purple. Okay, we're going to do the mordant the mordant step of the gram stain. Okay, we're going to add our iodine. It's going to make a complex with the crystal violet that will make it stick to the peptidoglycan layer in the gram positives because it's a thick layer, but it doesn't do the same. Well, it does complex in the gram negatives, but there's not as much peptidoglycan, so it can't really stick because of that. Okay, so we're going to let the iodine set for a minute. We're going to rinse it off. Okay, then we're going to go to the decolorizing step, where we're going to add the ethanol. That's going to be our alcohol that we're using. And this is going to wash away the stain in the gram negatives, but leave the stain in the gram positives. Now, with the decolorization step, with this ethanol step, if we leave the ethanol on for too long, what will actually happen is both the gram positives and the gram negatives will be colorless. Okay, so we have to really pay attention to how much time we have the ethanol step going for. Okay, because we can potentially decolorize the gram positives as well. So when we go and do the counter stain, both gram positives and gram negatives will appear pink even when they shouldn't. Okay, and the last step is our counter stain step. I'm not sure why this isn't bolded. It should be. This is where we're going to use the safranin to make the gram negatives pink. As I said, you need to know this vocabulary. You need to know these steps. You need to know what happens at each step. Okay, so this is what we're actually going to see. So our gram positives, this is a type of bacteria that we call firmicutes. You don't need to have that memorized yet. We'll come back to it in a later chapter and explain exactly what a firmicute is. But some classic examples of firmicutes are the Staphylococcus genus. So this is going to be Staphylococcus aureus that causes MRSA and staph infections, Staphylococcus epidermidis that we can find on our skin cells. We can also have Streptococcus genus. Okay, this is going to be the bacteria that causes things like strep throat. Okay, these are all going to be purple. Okay, because they have that thick peptidoglycan layer. Now, gram-negatives, these are what we call proteobacteria. So proteobacteria, we find it mostly in the gut bacteria. So this is going to be things like E. coli and Proteus bacteria. Okay, so things that we find in our gut are going to stain pink. Okay, the gram stain is so important to us. We do it in the lab, we talk about it in class. We talk about the gram stain even after this chapter when we first go through it. So what I'm going to do is I'm going to play a animation from the textbook website going through what I just told you all again. Okay, this is going to be something I want you to see several times because it's so important for class. And this is something that we have to be able to do, and this is something that we have to know the steps of for both class and lab.
A simple technique called the gram stain, combined with light microscopy, distinguishes two classes of bacteria, gram-positive and gram-negative. Here are gram-positive bacteria in a sample of sputum from a patient with pneumonia. The gram-positive bacteria have taken up and retained a dark purple dye. They are visible against the pink background of human epithelial cells. The second sample shows gram-negative bacterial cells, negative because they do not retain the dark purple color. The gram-negative bacteria are seen because they take up a secondary pink dye. The gram stain procedure is called a differential stain because it produces different colors for different kinds of cells. The gram stain distinguishes cells based on their cell walls. Just outside the cell membrane, a gram-positive bacterium has a thick cell wall, a rigid mesh-like structure with multiple layers of a molecule called peptidoglycan. In contrast, gram-negative cells have just one or two layers of peptidoglycan, outside of which they have a second membrane, called the outer membrane. These structures behave differently during the gram stain. Before using the gram stain procedure, cell samples must first be prepared. A loopful of a bacterial culture is smeared as a thin film on a microscope slide and allowed to air dry. The cells are then fixed or adhered to the slide by heat or by adding a drop of methanol, and again air drying. Here we'll follow the procedure as it would occur using fixed samples of gram-positive and gram-negative cells. The gram stain uses 4 different solutions: the bright purple dye called crystal violet, iodine, ethanol, and the red dye called safranin. Crystal violet is applied first and left on for a minute. Crystal violet is a positively charged dye. The positively charged group is attracted to negative charges inside and outside the cells, including to negatively charged cell walls. Excess stain is washed off with water, leaving cells from both samples colored. The iodine solution, added next, is left on for 1 minute. The iodine is a mordant, which is a substance that helps set a dye. Iodine complexes with the crystal violet, making it precipitate and become trapped in the cell and cell wall. The excess iodine is washed off. Next, a decolorizer, ethanol, is added for a precise interval, typically about 20 seconds, followed by a water wash. Ethanol removes loosely bound crystal violet iodide molecules, but gram-positive cells, with their thick mesh-like cell walls, retain the stain tightly. In the gram-negative cells, the ethanol disrupts the outer membrane, making the dye easy to wash away. In the final step, a counterstain, safranin, is applied. This process allows the visualization of gram-negative cells, which the safranin stains pale pink. By distinguishing between 2 major classes of bacteria, the gram stain became essential in the clinical laboratory. However, the gram stain is reliable only for freshly cultured bacteria. Additionally, some groups of bacteria, such as lactobacillus species common in the oral cavity and gastrointestinal tract, may stain either positive or negative and are thus not distinguished by the gram stain. They are gram variable.
Okay, so that's the gram stain. So what we're going to do to finish up chapter 3.4 is we're going to talk about some other differential stains. Now, you don't need to remember all the details about these, but you do need to remember their names and kind of how they work. Okay, so we're going to go through 4 other differential stains besides the gram stain. The first is the acid-fast stain. So we've talked about gram-positives and gram-negatives that have a difference in their cell walls and their peptidoglycan layer. So one has a thick peptidoglycan layer, the gram-positives, and one has a thin peptidoglycan layer, the gram-negatives. Okay, we have some bacteria that have cell walls that have what are called mycolic acids in them. So these type of bacteria won't gram stain very well, but they will do what's called an acid-fast stain. Okay, so this is going to use carbonyl fusion, and we're going to heat treat it to get the dye to get into the cell walls of these bacteria. Now, the acid-fast stain is used for things that have mycolic acid in them. So these are a lot of our mycobacteriums, mycobacterium, mycolic acid. Kind of see the difference here. Okay, so this is what we'd use to add contrast to the slides with Mycobacterium tuberculosis. Okay, so the bacterium that causes tuberculosis from our first case study with Debbie and her school. And then a different mycobacterium called Mycobacterium leprae. Okay, this is actually the bacteria that causes leprosy. Okay, so if we see here in our slides, so we can see the tissues, we can see these purple bacteria here. This is what the carbonyl fusion is going to give this purple color. And this is the Mycobacterium tuberculosis in the tissue. Okay, another one is our endospore stain. We've seen this when we talked about the case study about the pathogen in the can with Julio and his sister. Okay, so endospore stains are used for bacteria that have endospores. So things like the Bacillus genus and the Clostridium species. Okay, so Clostridium botulinum shows up really well with the endospore stain. Okay, so what's going to happen is we're going to take our sample and we're going to boil it with malachite green. Okay, so what this is going to do is the heat's going to allow the malachite green to get into the endospore. So this, like, you can think of it as this little ball that contains the genetic material of this bacteria, so the DNA. Okay, so what's going to happen is we're going to stain the outside of this endospore. We can't break it open. We need extremely high heat, and so like 120 degrees Celsius in order to kill endospores, right? This is why we have to use the autoclave. Okay, but we can boil it with malachite green and we can get that dye to stick on the outside. Okay, and what's going to happen is this is going to allow us to specifically see the endospore. So we usually use a counter stain like safranin, so get this pink color. So this blue that we see here is the endospore, and the pink's the rest of the bacterium. Okay, so this is looking at Clostridium tetani in a sample. Okay, so that's acid fast and that's endospore. Now the last two, so we have some bacteria that won't stain very easily at all. They may have cell walls, but they'll also have this kind of really thick and sticky capsule on the outside, or they'll have a lot of little polysaccharide filaments on the outside that don't allow staining to get in to stain the actual bacteria. And because this capsule and these filaments are preventing that, we have to approach this a little bit differently to view the bacteria. So we're going to use what is called a negative stain. Okay, so this is going to help us visualize these bacteria with the capsules and the filaments, because what it's going to do is it's going to stain the background or the area around the bacteria. So they're going to pop up as looking like these little white, in this case, rods. Okay, so this is how we can image them. See, this capsule is very important for these bacteria because it's going to make it so that our immune system can't grab a hold of those bacteria and engulf them to kill them. Okay, so it makes them kind of immune to our immune system, but it also makes them immune to our dyes, which is why we're going to use a negative stain. Now, I don't have any negative stained slides in the lab. I'm working on getting some. We will look at some micrographs like this one, to see what they actually look like, but I don't actually have some of these in the lab. And the last one we're going to talk about is the antibody stain. Now, we've seen examples of this in some previous case studies. Okay, so antibodies are very specific proteins for very specific molecules. So they can be proteins or any structure on a bacteria or a virus that these antibodies are going to stick to. Now, the beauty of the antibody stain is that the antibody stain is going to have the antibody, so the place that's going to bind, or sorry, the protein that's going to bind to, say, other proteins. And at the end of the antibody is going to have what we call a fluorophore. Okay, this is a molecule that is going to glow or fluoresce so that we can look at it underneath that fluorescent microscope and see it there. Okay, these are very specific because each antibody is only for a specific molecule. Okay, so when we use these, we're looking for a very specific bacterium that's causing a very specific disease. Okay, so what we see here is our antibody was for this bacteria, so it bound to it. And because it's bound to it, we see the fluorophore, or that glowing, underneath the microscope. If we didn't have that bacteria that antibody was specific for, we actually wouldn't see anything but dark underneath the fluorescent microscope, because there would be nothing for that stain to bind to. Okay, that is 3.4. This is a really important section. I would study it. This is going to show up a lot on your first exam, and this is going to be tested on in the lab portion of the class as well for your midterm. Okay, so we've talked about how to fix and stain specimen, okay, to improve contrast and resolution. We talked about differential stains, and most importantly, we talked about the most famous differential stain, the Gram stain, and how we can use it to tell the difference between gram-positive and gram-negatives. Okay, if we use the Gram stain on a eukaryotic cell, so like our cheek cells, which we're actually going to do in lab, our eukaryotic cells are going to stain negative, right? We don't have cell walls, we don't have a peptidoglycan layer, so there's nothing for the crystal violet and the iodine complex, crystal violet iodine complex to stick to. Okay, so eukaryotic cells are always going to be pink. Okay, so that's the end of 3.4. We'll see you in 3.5.